Today, we're exploring the key ideas from 10% Human by Alanna Collen. This is an independent educational overview enriched with our own integrative analysis and actionable exercises. Think of it as a guided reflection to spark your own insights, not a replacement for reading the book itself. Introduction: the unseen architect of our being. For centuries, humanity envisioned itself as the pinnacle of biological complexity, a self-contained entity whose intricate design resided solely within its own genes. The mapping of the human genome, however, revealed a humbling truth. Our genetic blueprint is surprisingly modest, far less extensive than once imagined, and scarcely more complex than that of simpler organisms. This revelation unveiled a profound biological paradox, hinting that the true wellspring of our complexity lay elsewhere. The missing piece, it turns out, resides in an invisible yet omnipresent dimension of life, the vast microbial ecosystem that cohabits every part of our bodies, particularly our gut. We are not solitary organisms, but rather intricate superorganisms, our own cells vastly outnumbered by a bustling metropolis of microbial partners: bacteria, fungi, viruses, and archaea. These microscopic inhabitants contribute an astonishing wealth of genetic information, dwarfing our own genetic endowment. This partnership, forged over eons of coevolution, underpins nearly every aspect of our health, from our immune defenses and metabolic efficiency to our cognitive functions and emotional states. The book meticulously uncovers how modern life, with its triumphs of sanitation, medicine, and industrial food production, has inadvertently waged war on these ancient microbial allies, leading to a cascade of chronic illnesses now defining the 21st century. It champions a radical shift in perspective, urging us to embrace our microbial inhabitants not as foes to be eradicated, but as indispensable collaborators whose well-being is intrinsically linked to our own. This is an invitation to redefine what it means to be human, acknowledging our profound and beautiful microbial interdependence. The inner cosmos: how microbes shape our physiology and fate. The human body, far from being a sterile vessel, teems with an astonishing array of microbial life, collectively known as the microbiome. This inner cosmos, predominantly housed within the gut, is a dynamic ecosystem whose genetic contribution vastly eclipses our own. We are, by sheer genetic diversity, more microbial than human. This profound symbiosis is not merely tolerated, it is actively cultivated, underpinning a myriad of physiological processes that define our health and even our personality. Disruptions to this delicate microbial balance are increasingly recognized as a root cause for a perplexing array of modern ailments. Consider the human immune system, a formidable defense network, often portrayed as a solitary guardian against foreign invaders. The reality is more nuanced. A significant portion of our immune tissue resides in the gut, where it engages in a constant dialogue with our microbial residents. These old friends, as they are aptly called, are not just tolerated, they actively train and calibrate our immune responses. Through evolutionary co-adaptation, beneficial microbes have developed sophisticated mechanisms to communicate with our immune cells, teaching them tolerance and preventing them from overreacting to harmless substances like pollen or even our own tissues. Specialized immune cells, acting as peacekeepers, learn to modulate inflammatory responses under the guidance of these microbial partners. When this microbial tutelage is lacking or compromised, the immune system can become hypersensitive, leading to the dramatic rise in allergies, where benign environmental factors trigger aggressive reactions, or to autoimmune conditions, where the body mistakenly attacks its own healthy cells, such as in certain forms of arthritis or nerve degeneration. The integrity of our gut lining is another critical interface controlled by our microbial partners. A healthy gut maintains a tight barrier, selectively allowing nutrients to pass into the bloodstream while blocking harmful molecules. However, an imbalanced microbial community, known as dysbiosis, can irritate the gut wall, prompting the release of specific proteins that loosen these protective junctions. This phenomenon, often termed leaky gut, permits bacterial byproducts and undigested food particles to seep into the bloodstream. The body's immune system, encountering these foreign substances where they don't belong, initiates a systemic inflammatory response. This chronic, low-grade inflammation is not merely a symptom, it is a driving force behind a spectrum of conditions, from metabolic disorders like insulin resistance and weight gain to various neurological and mood disturbances. For instance, heightened levels of bacterial toxins in the blood resulting from a permeable gut can contribute to the relentless storage of fat and the development of type 2 diabetes. Beyond metabolism and immunity, our microbial residents exert a startling influence over our minds. The gut-brain axis, a bidirectional communication highway, ensures that the state of our internal microbial ecosystem directly impacts our neurological and psychological well-being. Microbes produce a diverse array of neuroactive compounds, some mimicking human neurotransmitters, which can influence mood, anxiety levels, and even social behavior. Studies have revealed that altering an animal's gut microbes can literally change its temperament, transforming timid creatures into bold explorers, and vice versa. In humans, associations have been drawn between specific microbial profiles and conditions ranging from anxiety and depression to more complex neurodevelopmental disorders like autism. For example, certain gut bacteria can produce compounds that, when in excess, may contribute to behaviors reminiscent of autism, such as repetitive actions and social withdrawal, by disrupting neural development or causing neuroinflammation. The very structure and function of the developing brain appear to be profoundly shaped by the microbial world it encounters in early life. Even seemingly peripheral organs, like the appendix, underscore our deep microbial dependency. Once dismissed as a useless evolutionary remnant, the appendix is now understood as a vital microbial sanctuary. It acts as a protected reservoir, housing a diverse community of beneficial bacteria within a specialized biofilm. In times of intestinal distress, such as severe infection, this nest egg of good microbes can repopulate the gut, aiding recovery and reinforcing the immune system. The appendix's persistent presence across mammalian evolution speaks to the crucial, life-sustaining role these internal microbial partners play. Thus, our invisible passengers are not mere hitchhikers. They are integral architects of our physiological balance, mental state, and long-term health, blurring the lines between self and other in a profound redefinition of human biology. The unintended consequences of modern progress. The 20th century stands as a testament to humanity's ingenuity in combating disease. Revolutionary advancements in public health, from the eradication of scourges like smallpox through vaccination to the establishment of rigorous hygiene in medical practice, the widespread purification of water, and the life-saving advent of antibiotics, transformed human longevity. Average lifespans surged, and the fear of infectious disease receded dramatically in developed nations. Yet this triumph over acute pathogens has inadvertently paved the way for a new crisis, a surge in chronic, non-communicable 21st century illnesses that now afflict a significant portion of the global population. This paradox reveals the profound, often detrimental, consequences of our modern lifestyle on our unseen microbial partners. Foremost among these modern disruptors are antibiotics. Hailed as miracle drugs, antibiotics have undoubtedly saved countless lives. Yet their pervasive and often indiscriminate use has had far-reaching ecological consequences for our internal microbial landscapes. Beyond fostering widespread antibiotic resistance, which threatens to undermine their future efficacy, these powerful medications inflict collateral damage. They are broad-spectrum, indiscriminately wiping out beneficial microbial species alongside harmful pathogens. Even a short course of antibiotics can drastically alter gut microbiotic composition, with some species failing to recover for months or even years. This disruption, or dysbiosis, is linked not only to immediate side effects like digestive upset, but also to a heightened risk for a myriad of chronic conditions. The chronological overlap between the widespread introduction of antibiotics in the mid-20th century and the exponential rise in conditions like allergies, autoimmune diseases, e.g. type 1 diabetes, certain rheumatic conditions, and even obesity is strikingly compelling. Research demonstrates that early life antibiotic exposure in animals can disrupt metabolic hormones and increase fat mass, effects that can persist even after the drugs are stopped. Similarly, children with a history of frequent antibiotic use exhibit a higher likelihood of developing allergies and asthma, underscoring the critical role of early microbial education for a balanced immune system. Beyond pharmaceuticals, our societal embrace of hyperhygiene further exacerbates microbial depletion. The ubiquitous use of antibacterial soaps and sanitizers, often marketed as superior to plain soap and water, introduces harsh chemicals that indiscriminately eliminate beneficial skin microbes. These chemicals not only contribute to environmental pollution and antibiotic resistance, but can also disrupt hormonal balance and potentially increase susceptibility to certain infections. Paradoxically, by stripping away our natural microbial defenses, we may inadvertently create a vacuum that allows opportunistic pathogens to flourish, leading to a vicious cycle. The very measures designed to protect us from germs may be weakening our innate microbial resilience. Dietary shifts represent another critical assault on our microbial allies. Western diets are characterized by a drastic decline in dietary fiber, the complex carbohydrates found abundantly in plants. Our ancient human ancestors and contemporary populations adhering to traditional plant-rich diets possess gut microbiomes teeming with species specialized in fermenting diverse fibers. These microbes produce vital short-chain fatty acids, SCFAs, which nourish gut cells, modulate the immune system, and regulate metabolism. In contrast, modern processed foods, rich in refined sugars and fats but deficient in fiber, starve these beneficial fiber-loving bacteria. This leads to a less diverse, more resilient deficient microbiome, often dominated by species associated with inflammation and metabolic dysfunction. The global obesity crisis, for instance, cannot be solely attributed to increased calorie intake. Rather, it is intimately linked to a microbial community that, deprived of fiber, becomes less efficient at extracting energy and more prone to storing it as fat, contributing to systemic inflammation and impaired satiety signaling. Finally, the fundamental processes of human reproduction and infant development have become profoundly altered in ways that disrupt critical microbial seeding. The increasing rates of cesarean sections, while life-saving in many instances, bypass the natural microbial transfer from mother to infant that occurs during vaginal birth. Babies born via C-section acquire different initial microbial communities, often skin and hospital environment bacteria, rather than the diverse maternal vaginal and fecal microbes that are crucial for kickstarting a healthy immune system and metabolic programming. Similarly, the widespread use of infant formula, while providing essential nutrition, lacks the unique oligosaccharides and live beneficial bacteria found in breast milk. Breast milk is not just food, it is a meticulously designed microbial incubator, feeding specific beneficial bacteria and directly transferring maternal microbes to the infant's gut. These early life microbial disruptions, at birth and during infancy, are linked to higher risks of allergies, autoimmune conditions, and obesity later in life, highlighting how even our earliest moments are critically dependent on a healthy microbial legacy, now often inadvertently compromised by modern medical and lifestyle choices. Reclaiming our microbial legacy. The pervasive rise of chronic illnesses in the modern era, linked to a compromised microbial ecosystem, underscores an urgent need to reclaim our lost microbial heritage. While the problem is complex and deeply ingrained in societal norms, empowering solutions exist, ranging from targeted medical interventions to fundamental shifts in individual lifestyle and public health strategies. The central principle is clear. To foster health, we must actively nourish and protect our microbial allies. For severe, acute microbial imbalances, radical yet highly effective solutions are emerging. Consider Clostridium difficile infection, a debilitating and potentially fatal gut condition, often triggered by antibiotic use. For recurrent cases, conventional antibiotics frequently fail and can even worsen a problem. Here, fecal microbiota transplantation, the transfer of stool from a healthy donor into a patient's gut, has proven astonishingly effective. With success rates often exceeding 90%, FMT represents a powerful paradigm shift, recognizing that replacing an entire ecosystem of beneficial microbes can restore health where single-agent drugs cannot. This ancient practice, albeit modernized, highlights the profound impact of comprehensive microbial restoration, paving the way for future innovations like crapsules, oral capsules containing donor microbiota, and synthetic microbial cocktails, engineered to mimic a healthy gut community. However, the primary focus must shift from reactive treatment to proactive prevention and long-term microbial health. At the heart of this strategy lies dietary transformation. Our ancestors and contemporary populations free from the burden of modern chronic diseases consume diets exceptionally rich in diverse plant fibers. These fibers are not primarily for our human cells. They are the essential nourishment for our beneficial gut microbes. A diet abundant in fruits, vegetables, whole grains, legumes, nuts, and seeds cultivates a diverse and resilient microbial community, one capable of protective short-chain fatty acids, strengthening the gut barrier, and modulating the immune system. This means re-evaluating our relationship with carbohydrates, distinguishing between refined sugars that offer little to our microbes and complex, fiber-rich carbohydrates that are their sustenance. Even small, consistent changes, like choosing whole grains over refined, incorporating a wider variety of plant-based foods, and opting for less processed options, can profoundly reshape our internal landscape, encouraging the growth of beneficial species linked to leanness, immune resilience, and mental well-being. Crucially, we must fundamentally alter our relationship with antibiotics. While undeniably life-saving and indispensable for serious bacterial infections, their overuse and misuse has come at a steep microbial cost. This calls for a judicious, informed approach, reserving antibiotics for confirmed bacterial infections, questioning their necessity for viral illnesses, and exploring targeted diagnostics to minimize broad-spectrum exposure. When antibiotics are necessary, co-interventions like specific probiotic strains or strategies to actively replenish the microbiota afterward can help mitigate their collateral damage. This shift requires both patient advocacy and medical education, fostering a culture where antibiotics are respected as powerful tools, not indiscriminate panaceas. The earliest stages of life offer the most critical window for shaping a healthy microbial legacy. For expectant parents, choices surrounding childbirth and infant feeding hold profound implications. Prioritizing vaginal birth when medically appropriate ensures the vital transfer of maternal microbes to the newborn, seeding a foundational community essential for immune development and long-term health. In cases where a cesarean section is medically necessary, emerging practices like vaginal seeding, swabbing the newborn with maternal vaginal fluids, offer a promising avenue to mimic this crucial microbial initiation. Similarly, breastfeeding, far from being merely a nutritional choice, is an unparalleled microbial intervention. Breast milk contains not only complex sugars that selectively feed beneficial infant bacteria, but also live maternal microbes directly transferred to the baby. These components are absent in formula, and their absence is linked to higher rates of infections, allergies, and metabolic disorders in formula-fed infants. Empowering parents with this knowledge allows for informed decisions that prioritize a child's microbial future. Ultimately, reclaiming our microbial legacy is about recognizing our symbiotic nature. It means shifting our mindset from a germ-phobic approach to one of microbial embrace, understanding that health is not a state of sterility, but a harmonious coexistence. By making conscious choices about what we eat, how we use medicine, and how we bring up our children, we possess unprecedented power to cultivate a thriving internal ecosystem. This journey of microbial restoration is not just a personal quest for well-being. It is a collective responsibility to preserve and pass on this vital, invisible inheritance for the health and happiness of future generations. Integrative Analysis and Implications. The exploration of the human microbiome fundamentally redefines our understanding of health and disease, presenting a profound paradigm shift that challenges long-held anthropocentric views of biology. The central argument that humans are superorganisms, deeply reliant on a vast and diverse internal microbial ecosystem, is no longer a fringe theory, but a robust scientific reality. This new lens illuminates the ironic truth that the triumphs of 20th century public health, while eradicating many infectious diseases and extending lifespans, inadvertently sowed the seeds for the chronic ailments plaguing the 21st century. Our concerted war on germs has inadvertently decimated our old friends, leaving our immune systems disoriented, our metabolisms dysregulated, and our mental states vulnerable. The implications of this microbial revolution are far-reaching, demanding a comprehensive reevaluation across multiple domains. In medicine, the focus must shift from solely targeting human genes or individual pathogens to understanding and modulating entire microbial communities. This opens avenues for personalized medicine, where an individual's unique microbial profile could guide diagnostic strategies, predict drug efficacy and side effects, and inform tailored therapeutic interventions, including next-generation probiotics or customized fecal transplants. Developing rapid, targeted diagnostics for infections, for instance, would allow for more judicious antibiotic use, preserving these invaluable drugs for when they are truly necessary and minimizing ecological disruption. Public health strategies, too, require a microbial recalibration. Campaigns promoting diverse, fiber-rich diets, emphasizing whole, unprocessed plant foods, become as critical as vaccination programs. Reevaluating hygiene standards to differentiate between pathogenic threats and beneficial microbial exposure is essential. Policies supporting vaginal birth and breastfeeding become not just recommendations, but vital investments in the lifelong health trajectories of new generations, acknowledging the profound microbial inheritance passed from mother to child. Education at all levels is paramount, moving beyond simplistic germ theory to foster an understanding of symbiotic coexistence. At a societal level, this perspective challenges us to reconsider our relationship with the natural world. The loss of microbial diversity within our bodies mirrors the broader ecological crisis, highlighting the interconnectedness of all life. Embracing our microbial partners means acknowledging that we are part of a larger, interdependent web, and that our internal ecosystems thrive when external ecosystems are healthy and diverse. This necessitates a more thoughtful approach to agriculture, food production, and environmental conservation, recognizing their downstream effects on the human microbiome. Individually, the insights from microbial research offer a profound sense of empowerment. While we cannot alter our inherited human genome, we possess significant agency over our microbiome. Dietary choices, lifestyle habits, and informed decisions about medical interventions become powerful levers for shaping our microbial partners, and by extension, our health. This encourages a shift from passive patient to active participant in one's own well-being, fostering a deeper, more mindful engagement with food, medicine, and the environment. The book's journey, culminating in the author's personal transformation, vividly illustrates this empowerment. Her experience underscores that restoring microbial diversity and function can lead to tangible improvements in chronic conditions, demonstrating that cure is not merely the absence of pathogens, but the presence of a flourishing, balanced internal ecosystem. Ultimately, the microbial lens offers a hopeful vision. By recognizing and nurturing the 100 trillion old friends within us, we can unlock a deeper understanding of health, resilience, and happiness, moving towards a future where humanity lives not just longer, but truly more fully, in harmony with its fundamental biological nature. We are, indeed, only 10% human, and our true selves emerge from this magnificent partnership. Actionable Exercises. -1. Fiber feast for your friends. Your gut microbes thrive on diverse plant fibers. Think of your plate not just as food for you, but as a delicious meal for your trillions of tiny partners. By nourishing them, you're nourishing yourself. Identify 5 to 7 new plant foods you rarely or never eat. Examples: different types of legumes, root vegetables, unusual fruits, less common whole grains. Incorporate one new plant food into your diet each week, starting with small portions. Focus on a wide variety of colors, textures, and types from all plant categories. Observe how your body feels. Notice changes in energy levels, digestion, or mood. This helps you understand how you're feeding your internal ecosystem. Chapter 2. The Thoughtful Antibiotic Check-in. Antibiotics are powerful, life-saving tools, but their widespread use comes with a cost to your beneficial microbes. Becoming more mindful can protect your inner ecosystem. When facing an infection, take a moment to confirm with your doctor if it's bacterial or viral. Remember, antibiotics are only effective against bacteria. Discuss with your doctor if antibiotics are truly necessary, or if a wait-and-see approach or alternative treatments are viable for minor ailments. If antibiotics are essential, inquire about the narrowest spectrum drug for the shortest effective duration. Support your gut during and after a course by consuming diverse fermented foods and discussing specific, evidence-based probiotics with your healthcare provider. 3. Cultivating your inner and outer microbial garden. Our ancestors lived in close contact with diverse microbial environments. Reconnecting with nature and reducing excessive sterilization can reintroduce beneficial old friends to your body. 1. Embrace nature. Spend time outdoors daily, gardening, walking in a park, or hiking. Direct contact with soil and diverse natural environments exposes you to a wider array of beneficial microbes. 2. Rethink cleaners. Reduce the use of harsh antibacterial soaps and sanitizers for routine cleaning. Simple soap and water are often sufficient and help preserve your natural skin microbiome. 3. Befriend fermented foods. Gradually introduce naturally fermented foods into your diet. Example: sauerkraut, kimchi, kefir, yogurt with live cultures, kombucha. Start small to see how your body reacts. 4. Connect with companions. If possible, interact regularly with pets, as they can introduce different microbial diversity into your home and life. 4. Decode your body's microbial messages. Your gut is a crucial communication hub, deeply connected to your immune system and brain. By paying attention to your body's signals, you can learn to understand what your microbial partners might be telling you. Start a microbe journal. For 2 weeks, track daily digestive comfort, bloating, regularity, energy levels, mood shifts, anxiety, focus, and any persistent skin issues or general discomfort. Note inputs. Alongside your symptoms, record what you've eaten, any new medications, significant stressors, or changes in your environment. Identify patterns. Look for correlations. Do specific foods consistently trigger digestive upset or mood changes? Does stress impact your gut? This helps you connect your lifestyle choices to your internal microbial world. Listen to your gut. Use these insights to make small, informed adjustments to your diet or routine and observe their impact. 5. Building a strong start. Microbial legacies. The first few years of life are critical for establishing a child's foundational microbiome. For current or future parents, informed choices during this period can set the stage for lifelong health. 1. Research birth options. Understand the microbial benefits of vaginal birth for a baby's foundational microbiome. If a C-section is medically necessary, discuss potential interventions like vaginal seeding with your healthcare provider. 2. Prioritize breastfeeding. Learn about the unique microbial and prebiotic components in breast milk that nurture a baby's developing microbiome. Plan for extended breastfeeding according to WHO guidelines if possible. 3. Create a less sterile home. Avoid excessive use of antibacterial products in your home environment. Encourage children to play outdoors and interact with nature and pets within reasonable hygiene limits to foster diverse microbial exposure. That wraps up our exploration of today's book. If even one idea here gave you something to reflect on, give it a like so YouTube knows to share more with you. And if you'd like the next insight in your feed, hit subscribe. Remember, what you heard today is our independent interpretation, enriched with analysis and exercises. The full book has far more depth, so consider reading it to experience the author's work directly. Thanks for listening to DeftU Insights, where we turn great books into tools for growth.